Okay, thank you very much for that. If I hadn't recorded it when you just asked me to, that would mean I'd have to sit here tonight and record it all to myself. Uh, that's not fun. Okay, so we haven't started anything yet. Uh, uh, so there's nothing to backtrack on. We got it just in time. Thank you. Okay. This introduction paragraph says that every stimulus from the peripheral nervous system, that's your arms, your legs, everything except the spinal cord and the brain. Every stimulus that comes from the PNS has to reach the CNS to have any effect. Okay, if you're going to elicit a response, it's got to make it to the central nervous system. The central nervous system determines what, if anything, to do about the stimulus. The exception in parentheses here at the end of this paragraph is that this isn't going to hold true for sight, sound, taste, and hearing, because the nerves for this are located in the head. Okay, these are cranial nerves. The nerves of the peripheral nervous system have to enter the CNS, and they do it through the spinal cord. Okay, they do it through spinal nerves. In this drawing or illustration, the spinal nerves are in yellow. Is that yellow? Close enough?
Yes, it's
Yes.
Okay. And you see where they enter in between vertebrae. If you may have observed this, although I did point it out, long ago when we were doing the vertebral column and the bones, there was space left. And that's where the peripheral nervous system, by way of spinal nerves, enters in. There are 31 pairs of spinal nerves. If you're able to count down, they're probably not all in the illustration, but here's the lumbar region and here's the sacral region. There would be 31 pairs. There are 8 pairs of cervical spinal nerves. Now, if you count here, I got 1, 2, 3, 4, 5, 6. You're not seeing 7 and 8. They're there. Okay, so you take it from the text here that there are 8 pairs of spinal nerves. How many cervical vertebrae are there? Think back. How many vertebrae in the neck?
7.
7. Thank you. We start with C1, that's the atlas, C2, the axis, and go to C7, the vertebral prominence. The first vertebra, C1, has a spinal nerve pair enter above it and a second pair enter below it. All of the rest have a spinal nerve pair entering below it. Okay. So there are 7 cervical vertebrae, all of which have spinal nerve pairs entering below them. But C1 has an additional set of spinal nerves entering above. That gives a total of 8 here. Now, what are spinal nerves? We'll get to that just a second. Just hang on, okay? In the thoracic region, there are 12 pairs of spinal nerves. 1 pair of spinal nerves under each of the 12 thoracic vertebra. In the lumbar region, one pair below each lumbar vertebra, and since there are 5 lumbar vertebrae, then there are 5 pairs of spinal nerves. In the sacrum, even though in the adult we consider it one bone, during its formation, it's a fusion of 5 bones, and a spinal nerve is underneath each section. They end up protruding from those sacral foramina that we saw earlier. Since there are 5 sacral regions, I'll call them, okay, you have 5 pairs of spinal nerves. And the coccygeal region has 1 pair of spinal nerves itself. The total number is 31. Okay, these spinal nerves are your connection or interconnection between the outside and the inside. The outside being the world of the PNS, and the inside being the central nervous system. We must have a connection. As it says a little bit earlier, if we don't have the connection between the PNS and CNS, nothing can be accomplished. These nerves are relatively short. You don't see a lot of yellow stretching for a foot or so. You just see maybe a fraction of an inch. Okay. Now, we looked at this before. We need to look at it again to know that you've got it back in your mind. This is a model of a vertebra. It just so happens that it's a cervical vertebra. We would know that 2 ways. One, the spinous process is divided. It's not always, but when it is, it's always a cervical vertebra. The division of the spinous process is referred to as being bifid. We also see that there are vertebral foramina here, here. You're not seeing the hole. You're seeing what's in the hole, however. You're seeing here and here vertebral arteries. The vertebral artery that we looked at for the last practice. But there's a hole here and a hole here. Okay, so this is in the cervical region, and most of it pertains to the entire spinal system. Okay. When an impulse from the peripheral nervous system comes in, okay, it comes in by the afferent pathways. Afferent meaning coming in towards the central nervous system, and it passes through this spot right here. This is a spinal nerve. We have another spinal nerve unlabeled on the other side, of course. Comes in, and then the incoming sensory stimuli enter into their parakaryones, or cell bodies, and form a dorsal root ganglion. These incoming impulses are coming in by pseudo-unipolar neurons. These are the neurons we said have a long axon in, then a swelling, and then they continue on. All the swellings, which are the cell bodies, accumulate here and make this enlarged area, which is a ganglion. A ganglion is an accumulation of neural cell bodies that is located in the peripheral nervous system. And we are still in the peripheral nervous system here. I haven't hit the spinal cord. Okay, so coming in, spinal nerve, dorsal root ganglion, following the dorsal root and then entering here at the dorsal horn. Okay. A little repeat from what we said on Tuesday as well. Okay. The information can now go to the brain, can also make a reflex arc. We won't worry about which it does yet. Here's the dorsal horn on this side. Here's the ventral horn. This material here is gray matter. It's all part of the CNS now because it's part of the spinal cord. And the white matter here, the white matter is myelinated by oligodendrocytes. Now I'm probably saying words you're going, wait a minute, you mentioned that last lecture. You mentioned it 2 lectures ago, but I don't remember it. I try to repeat as often as I can, but if you get lost, you need to say something, especially in today's lecture. Okay, incoming information comes in through the dorsal side by the dorsal root, enters the dorsal horn, may now go up to the brain, come back down. When it comes back down, it doesn't necessarily have to stop here. It can go all the way to the final section of the spinal cord and stop and get off at any point. What gets off? The impulse that's being sent out by the brain. Okay. Gray matter is not myelinated. It moves a little bit more slowly. Gray matter not myelinated. White matter is myelinated. Okay, we've taken care of all of the arrows in this drawing, but we can now look at the one below. Here's the spinal nerve again, coming in. Okay, dorsal root ganglion unlabeled. Here's the horns, dorsal, ventral, and then outgoing motor impulses leave here. And this is the ventral root. It's not labeled here, it is a little bit later on. The ventral root is efferent, efferent meaning going out. You see a piece of the ventral root here, the tip of the pointer, and it goes back out, and damn, it goes through the spinal nerve too, going the opposite direction. Spinal nerves are always mixed nerves. Incoming, outgoing. Okay. Two-way traffic, two-way traffic. This model shows an additional structure that we did test on. And today we're going to make some sense out of it, I hope. The sympathetic trunk ganglion. Okay. This sympathetic trunk ganglion, first it's a ganglion because look at it, the swelling, the accumulation of synapses and cell bodies that are in here cause it to swell. There's a connection to the spinal nerve that brings impulses towards it. This is the sympathetic trunk ganglion, meaning it's only being used by the sympathetic nervous system. The parasympathetic nervous system will have nothing to do with this. Again, you have to hold off until you've got the entire story. This is a drawing that I put together, and it's a typical thoracic vertebra. Now, a moment ago, I did the cervical vertebra. This one is a thoracic vertebra. Okay. What's the difference? Well, we're going to see it. First off, the way in from the peripheral nervous system is by following these red arrows. Or are they green? These arrows bring incoming or afferent impulses. And they do it from these two structures here. This is the primary ventral ramus. You do not need to know that term. This is the primary dorsal ramus. You do not need to know that term. And it tells you that down here in the text. It's just two main ways in. Okay. Ramus just means arm anyway. Ventral towards the belly. Dorsal towards the back. So you're looking at a person here, okay? And the back of the person is away from you, and the front is here. We bring in the impulses. Let's make up one. Pain, that's a good one. You just had, now I was going to say inoculation, but I'm tired of inoculation. You stabbed yourself on a thorn. Okay, you have pain sensation coming in. It will now go through this structure, and this is the spinal nerve. Not very long, and still coming inward. The sensation travels through the dorsal root ganglion, and then by way of the dorsal root to the dorsal horn. We have white matter labeled here again. Here's the ventral horn. And this time we have an enlargement on each side of the horn. This is the lateral intermediate mass. That'll be important in just a moment. The impulse travels possibly to the brain, or sometimes makes a direct reflex arc and comes straight across to the ventral root. In any event, for a command from the brain or any other part of the central nervous system to leave and go out to the peripheral nervous system to have an effect, it must leave through the ventral root. Dorsal root in, ventral root out. Here goes the ventral root. It combines with the dorsal root and runs side by side in the spinal nerve, which is again a mixed nerve. And from here, that impulse can run out backwards to the body. If this is skeletal muscle that's being innervated, then we're talking about the voluntary nervous system. The voluntary nervous system in reality is a very difficult subject, but on paper, like for us, very simple. It's bring an impulse in, evaluate it, send an impulse out, and send it back either to where it came from or to some other spot, higher or lower, to affect maybe a different muscle. Okay. If you, for instance, have a bee on your right arm, you slap it with your left arm. Okay. So you're going to employ a different part of the body. Okay. But just in and straight back out, voluntary nervous system. But we need to talk about the autonomic nervous system. And that's going to be a little bit different. It's going to involve these structures. This is a sympathetic chain ganglion. It's also known as a sympathetic trunk ganglion. Okay, this is what we saw just a moment ago, right here. Notice how this one goes up and it's cut off. Well, mine goes up and it goes up to the next vertebral level. And there's another chain ganglion. And another one above that until we get up to C1. And then there aren't any more. And it goes down as well. This probably reminds you either of a necklace or a chain where the beads clip together. This is a trading post of information and pathways that the autonomic nervous system will use. What do I mean? You can have autonomic nervous system commands coming from the spinal cord as well. So far, a moment ago, I just mentioned voluntary nervous system, but the involuntary nervous system, the ANS, also initiates from the spinal cord. And to leave the spinal cord, it'll take the ventral root as well, through the spinal cord. And then it has a possibility of going in this or this channel. It's the second channel that it can go into. The second channel is the white ramus. And it goes right into the sympathetic chain ganglion. There, that first sympathetic neuron, the one we call the preganglionic neuron on Tuesday, it can synapse with the postganglionic neuron. And the postganglionic neuron can take a turnaround and leave by this channel and now go to an organ that's going to be innervated by the sympathetic nervous system. There's an in and an out in the sympathetic nervous system. The in is the white ramus, and the out is the gray ramus. The only way to get in is to have the white ramus. You cannot go inward with the gray ramus. You can only go outward with the gray ramus. White and gray. Where did we hear that before? We heard white matter and, not labeled here, gray matter. Okay. The white ramus is myelinated, and the gray ramus is not, and it has the postganglionic neurons in it. Okay. This drawing is a typical thoracic vertebra, as it says at the top of the page. The thoracic area is part of the thoracolumbar or sympathetic nervous system. So this only pertains to the sympathetic nervous system of the ANS. This drawing does not pertain to the parasympathetic nervous system. Okay. So what does it pertain to? All of the thoracic vertebra and all of the lumbar vertebra. So no matter what level I wanted to draw this at, it would look essentially just like this for the thoraco and lumbar areas. Yeah. Not for the brain, not for the sacrum. We have that drawing coming up. Now, as I scroll past, you're probably feeling I don't know what to say. I don't get it yet, and I don't know how to ask a question about it. Give it a moment. Give it a moment. Okay. Same drawing. Same drawing. You might as well think that this is in the lumbar region or the thoracic region. Okay. Here's where the preganglionic neuron starts. Remember, the ANS, whether it's sympathetic or parasympathetic, the motor neurons travel through two nerves or two neurons, just two. The first is the preganglionic, the second is the postganglionic. Okay, the neuron for the preganglionic neuron, the cell starts here in the swelling that we call the lateral intermediate mass. Okay. This is the junction then of the CNS, the spinal cord, and the ANS. This is exactly where it happens. So now the incoming information, the stimuli, that's all the same. That's all the same. It's coming up through the spinal nerve, the dorsal root ganglion, dorsal root, go to the brain. Now, some of that action coming back, initiated by the brain, is going to go to the voluntary nervous system, but some of it will go possibly to the autonomic nervous system, and that portion starts here. And in red, here's the preganglionic neuron, leaving by the same way, the ventral root, going behind here. This is where the spinal nerve is. And it goes forward, passes by the gray ramus, because nothing can go into the gray ramus. Goes to the next one, and this is the white ramus. And it enters in and goes to the sympathetic chain ganglion. Okay. At that point, it can synapse with the undrawn, as yet, undrawn postganglionic neuron. That postganglionic neuron can turn around and go out this way through the gray ramus. That postganglionic neuron can also go up or down and exit at another level of the body. Now remember, these are actually nerves that are coming through, and they are composed of thousands of neurons. So you can take one nerve and split it up to 1,000 ways. It won't be that many, but you can split it. Part of the nerve and its information can go upward, and part can go down. It's amazing overkill. Here's another thing that can happen. The preganglionic neuron is initiated, goes out through the ventral root, spinal nerve, white ramus, but it doesn't stop there. It itself immediately travels up or down the chain and goes to another chain ganglion where it might synapse. It can synapse there. It can actually split also and actually go both ways and synapse. What's the idea here? We're taking an autonomic nervous system command, and we're splitting it up throughout the body. The autonomic nervous system and the sympathetic system is the one for fight or flight. And it's a good idea in fight or flight, get ready, to take the command, a single command, bring it down and split it every which way you can. and get the entire body primed. Same drawing. Preganglionic neuron. Okay, coming in, or coming out, I should say. This is the ventral root, spinal nerve, follow the red, go down through the white ramus, synapse. This one's going to synapse right here. And the postganglionic neuron in blue is going to exit by the gray ramus and come around. Why not just go straight across? Why not just skip this? Because if you skip this, then you skip the opportunity to have synapses form that direct the impulse up and down the body. Not all of them go up and down the body, but you want that opportunity. You want that opportunity, especially in the sympathetic nervous system. This again is just the sympathetic nervous system you're looking at. Now, don't have to worry about this little guy here. This is a collateral ganglion. Collateral to what? To the chain ganglion. Okay, they're not as regular. There's one for the heart. There's one for the celiac region. Okay, we're not going to go there. Not yet. The white ramus always conducts impulses into a sympathetic trunk ganglion. The gray ramus conducts impulses out of a sympathetic trunk ganglion back into the primary ventral ramus. I've written it out here, but I told you a moment ago, and it's in the text. You don't have to remember that term. Okay. Now, the chain ganglia that you see here run all the way from C1 down to the other C1. What's the other C1? Coccygeal vertebra 1. There's a chain ganglion for all of the vertebrae that we have and for all of the 31 spinal nerves. 31 chain ganglia on one side of the body and 31 on the other side. So there really is a total of 62 of these guys. Okay, we haven't drawn the other side. That means from the sympathetic region, which is the thoracic and lumbar region only, we can have this occur here, where the autonomic sympathetic command reaches the chain ganglion, and from there can go up or down, all the way to the top of the vertebral column or all the way to the bottom of the vertebral column and excite the entire body. That's not going to be the case with the parasympathetic pathways. These emerge from the brain and the sacrum or sacral region. I'm going to show you in picture what this is saying in text. This is a cross section through the sacral region. Just like in the rest of the vertebral column, the spinal cord column, we have a lateral intermediate mass, a swelling. And that's where the preganglionic neuron for the parasympathetic system arises. That's fine. It arises from the same location as the preganglionic neuron for the sympathetic system. But as you remember, the sympathetic system, let me back up, has a short, relatively short preganglionic neuron and a long postganglionic neuron. We don't even see the end of it. It's running all the way out to the organ. In the parasympathetic system, the preganglionic neuron is long. Here it is, going out through the ventral root, skips by this. Well, this is a gray ramus. This is a gray ramus, and it's sending information out. And that information coming out of the gray ramus would be from the sympathetic system. Just a moment ago, we said that the sympathetic commands can go all the way from the top to the bottom through these chain ganglia. The parasympathetic commands cannot, because there's no white ramus, there's no way to get in. There's no white ramus anywhere except in the thoraco or lumbar region. And without a white ramus, these commands from the parasympathetic system cannot reach the sympathetic trunk ganglion at all. Instead, they continue on and they find a long pathway called a splanchnic nerve. The splanchnic nerve runs right to the organ with just a fraction of a millimeter, possibly, between the end of the splanchnic nerve and the organ. And what fits in that little tiny space is a little tiny neuron called the postganglionic neuron. So parasympathetic pathway starts with a long preganglionic neuron that doesn't participate in the sympathetic trunk ganglion. After the long neuron, there is a short postganglionic neuron, and then the myo-neuro junction. So long as an impulse is in the chain ganglia, and those chain ganglia are located in the thoracolumbar region, there will be a white ramus. Okay. But there isn't anywhere else. All chain ganglia have a gray ramus. All 31 on each side have a gray ramus, meaning that the information coming to the sympathetic trunk ganglion, which can only get there from the thoracic and lumbar regions, that information can go to the sacral region and the coccygeal region, and it can go up to the brain. But it'll also bypass the cervical vertebra. The cervical vertebra do not have a white ramus either. The only ones with the white ramus to send information to the sympathetic trunk ganglion are the thoracic and lumbar regions. When we think back to the cervical region, let's see if I've got one. Well, that's not cervical, that's thoracolumbar. Okay, when we go back to our original picture here, we know that this is a cervical vertebra. What does the cervical vertebra have to do with the autonomic nervous system? Is it part of the sympathetic or parasympathetic system, both or neither? This time I am going to wait for an answer.
Neither.
Neither is the answer. Okay, neither. Okay, we have thoracolumbar, and we have craniosacral. We don't have anything in the ANS that's called cervical. But the cervical region does have chain ganglia. Okay, those chain ganglia receive their information, their commands from the sympathetic system, and they can exit those commands here. So there's only one showing. This is a gray ramus. The body and indirectly the mind through the thoracolumbar region can tell the cervical region what to do, but it will never be told what to do from the cervical region because there's no white ramus, and these vertebrae do not have the ability to enter into the autonomic nervous system. Okay, so don't be confused by this thing that sort of looks like a marine mine, for a boat or something like that. There's 31 of these in a line. 31. Okay, all of them have gray rami. The only ones with the white rami are the pairs of white rami that are in the thoraco and lumbar regions. This last little paragraph just repeats what I just showed you. There's no cervical autonomic nervous system input because there's no white ramus. Okay. Now there is autonomic nervous system input for the parasympathetic system, even though there's no white ramus, because they skirt that by using a splanchnic nerve, a long, dedicated nerve, dedicated to the organ that it's headed towards. That means that the information from the 5 sacral vertebra, okay, cannot be shared from one level to the next. It goes straight to the organ itself. There's no intermingling of commands between level 1 and level 2 and level 3. We don't have that here. Okay. If I ask for questions, I won't get any. I'm guessing. And that's usually the case when I go through it one time. Okay. But I will probably go through it again next week, probably on Tuesday, because I don't want to wait till after Thanksgiving. It'll be forgotten by that time. So. Okay, if you have any questions, hold on to them. If you dream up a question or think of a question, let me know in a moment when we come back. If you have, if you're confused, I'd like to hear that too. You haven't yet done the reading portion of it, I doubt, unless you happen to see that it was posted a couple days ago. Okay. But I need to know whether you what condition you feel you're in. You're going to get it. You're never going to get it. Which is it? Okay. So it's 3:06 right now. Let's break till 3:20. See you there. Okay. Okay, anybody with a question, comment, concern?
I have a question.
Sure.
The table that we're supposed to fill out, for the functions and stuff, are we gonna turn that in?
Yes,
Okay, when is that due again? By
P4.
P4, okay,
Yeah.
thank you.
I want you to keep it. You can turn it in early, if it's a copy, sure, you're done. You can turn it in, but obviously you want access to it uh, right up until the exam. Okay.
Okay,
Yeah. Okay. Okay. I'm gonna take you back to the website. You will have use of this. There's no need of repeating all this information that took years to, some of it took years to put together. But I want to point out something to you on nervous system. Okay. If you, the cranial nerves are repeated there. I'm not asking you to go there. The autonomic nervous system, that material, you have it on your module already. Okay. But way down at the bottom, I long ago put 4 parts of additional material on nerves. You don't need it for our class, but you may want to use it when you take physiology. These are notes from the physiology instructors, and I have unabridged notes, and then I've abbreviated those same notes so that you can see them in a different fashion. And I'm going to look at the last one. I think that's where I want to go. Number 4. If it's not, I'll find it. Okay, a lot of this is just the different format of things that we said. We have gone over all of this. We have actually gone over almost all of this. Maybe 1 or 2 things I didn't label. Okay, yes. The very last one, unabridged. You just saw this a moment ago. Okay. Well, I don't know which instructor you're going to end up with in physiology, assuming you're taking it here at Saddleback. There's more than enough of them right now. The one that is here and has been here the longest is Kara Street. And all of the rest of them, I believe, learned their material, got their material from her. So if that's indeed the case, or if certainly it is, if you're taking Kara Street, you're going to get hit with this. She's going to have a diagram on the board, and it will not have the preganglionic neuron in here like I had. And she's going to call since it's going to be in person, she's going to call on somebody. Could be you. And she's going to hand you a marker. And you're going to die like anybody else would with stage fright. And you're going to get up there and she's going to say, I want you to show the pathway of a preganglionic sympathetic fiber. And you're to draw it. So you would be drawing this in. Okay, if you hadn't reviewed this or listened to her notes carefully, you might have trouble doing that. But here is the answer. Another question she might ask, here is the answer. A third option, here is the answer. And the fourth option, the answer, the answer, the answer. Okay, so when you get up there and you know all the answers ahead of time, you've learned it, okay, and you will, and don't say that you got them from here. First, you may shock some of the other students into thinking, I better get busy. Okay, and that's great because usually do need that sort of push. You're going to impress her, and you shout out that you learned it all in Charlie's class. And but there they are. I'm just showing you. You don't need to go there for our test. Okay, the materials for our test and the practice test that I'll be putting up this weekend. Okay, those are all in our material, of course. Let's see. Oh. There's where we were, so I want to go back from that. Okay, today for lab, it's in two parts, but the two parts are very short and barely total even one part together. So we're also doing, in the lab, we're doing histology, of course, and we're also doing organs and organs systems. So you have to learn the organs, most of which we have covered before, as we've struggled to reach this point, and it should not give you too much difficulty. I'll hit that first one. Okay, I'm sure that if you go back to the digestive system and other topics that we've covered, you're going to find some duplication here. Well, duplication doesn't hurt. We'll call it review. Okay, but a lot of it, most of it is new material. In the digestive system, the first thing that we look at are the salivary glands. Okay, here they are in this picture here at the jaw. The largest salivary gland is the parotid, and it's drained by Stenson's duct, also known as the parotid duct. On a multiple choice test, I'm not going to put both of those. I'll put one followed by the second one in parentheses so that you're not confused. Here's the submandibular, also known as the submaxillary gland, and not shown because they're underneath the tongue and quite small and numerous, are the sublingual glands. Salivary glands secrete saliva. Saliva breaks down salivary, through salivary amylase, it breaks down starch into sugar. Does a poor job of it because we don't keep the material in our mouth long enough. And the acidity of the stomach denatures the enzyme. So it works for a short while. The main function of all of the salivary glands is to lubricate the food being swallowed. The pharynx The pharynx goes actually from behind the internal nares of the nasal passage all the way down to the larynx. Okay. Now, I have to sometimes ask, did I show you this picture before, or have I been just doing this too many years and it just seems like yesterday? Have you seen this one before?
Yes. Yes.
Okay, then we can go through it fast. Like I said, we should be able to consider some of this review. All of it's going to be on the exam, okay? And it's going to be on the final day, so we can imagine that final exams would contain some of our more distant material. The vestibule. The vestibule is the area in front of the teeth and behind the lips. Put the toothbrush in your mouth and polish the front side of your teeth. You have the toothbrush in the vestibule. The nasal pharynx is behind the nasal nares, so higher up. Anytime you gag on water or something that you're drinking or something that you've eaten and it goes up there, that's always the problem with the nasal pharynx. Uvula, the portion of the roof of the mouth that the soft palate hangs downward. Its function is said to be to close off the nasal pharynx when you swallow. Obviously, it doesn't work very well or all that well, or we wouldn't gag. Okay. At the back of the mouth is the oral pharynx. And here, right next to it, is showing the palatine tonsil. The palatine tonsil is the first thing I want to mention as far as tissue type is concerned. We'll overlap everything to get everything in there as many times as possible. The palatine tonsil is a lymph node, so it fights infection and is a place for white blood cells to accumulate, especially during times of infection. The tissue that makes up the lymph node in all lymph nodes is reticular tissue. Reticular tissue looks sponge-like, has cavernous areas for blood cells, especially white blood cells, to accumulate. The laryngopharynx is right here at the entrance into the trachea. That entrance can be closed by this structure produced made of hyaline cartilage. This structure that I'm touching, small, this is the epiglottis, and it will clamp down, shut down when you're swallowing something. The epiglottis is here. The epiglottis, the upper epiglottis anyway, is composed of stratified squamous epithelium. The lining of the trachea, we're not pointing out here, but it's available, is going to be an epithelium of pseudostratified ciliated columnar epithelium. We find that in the respiratory system, and that includes the nasal cavity. Pseudostratified ciliated columnar epithelium. This throat dissection shows the trachea quite well. The little rings that actually are not complete on the posterior side, these are the cartilaginous C rings. They're composed of hyaline cartilage. You have to use the probe, reach underneath the trachea to pull up the esophagus. The stomach we've gone over, I know, enough times. Really, I think we can just skip this one actually, unless there's a question. Just keep in mind, lesser curvature here, greater curvature here, and what's attached to each. The greater curvature has the greater momentum attached. The lesser curvature has the lesser momentum attached to it. In reality, when I see this model in the classroom, I cringe. I don't like it. I love this part where I'm arrowing now, but I don't like this part up here where they take a piece of liver and just cut it off. That's supposed to be liver with the gallbladder attached. The gallbladder produces bile, okay, and that bile helps in the emulsification of fat, as we all know. Exiting the gallbladder or entering is by way of the cystic duct. The hepatic duct is not labeled here. That would bring bile from the liver either to the ampulla of Vader or up into the gallbladder for storage. The stomach's been removed here, but here's the first part of the small intestine, the duodenum, and inside it's ribbed. The ribbing is the plicae circularis. The plicae circularis help to move the contents along, but more importantly, they increase tremendously the surface area of the lumen. And so with greater surface area, greater digestion, greater absorption. Here's the pancreas. Pancreas is a gland, and therefore it's made up of glandular epithelium. The adrenal glands are shown here, and they're made up of glandular epithelium. Now, we know that both of the organs have completely different functions, but the glandular type or the tissue type is always referred to as glandular epithelium. Both of these are merocrine glands, and so is the salivary gland that we passed by a moment ago. Merocrine gland means that it secretes a fluid, and that's it. The fluid can contain enzymes and anything else, but it can't contain cellular fragments, and it can't contain whole cells. This picture, I don't know if I gave it to you on P3, but it'll show up somewhere in the class. At first, it takes a little bit of getting used to. You see the ribs up here, and then you can convince yourself that that's the upper portion of the body, and then down here of the small intestines. Okay, and here is the stomach. The exit from the stomach is right here, and this is the cardiac sphincter. I'm sorry, the pyloric sphincter. Not thinking. The pyloric sphincter is the smallest diameter passageway in the GI tract. If anything can get through there, it can get through the rest. Here's the first part of the small intestine, the duodenum. I don't know why it's colored strangely like this. It usually is one color, this being the tan down here. Okay. Why this is dark, I don't know. Looking at a villus, it's one of these three. They're all villi. They protrude into the lumen of the small intestine only. There is no villi anywhere else in the body, only in the small intestine. They greatly increase the surface area again of the lumen, and they're there for absorption and digestion. Their outer layer is said to be the epithelial layer, and it is composed of simple columnar epithelium. All parts of the GI tract, with the exception of the upper esophagus, are simple columnar. The upper esophagus is stratified squamous. I grant you that this model does not do justice to the form that it should if we're looking at simple columnar. But since you know that this is the small intestine, has to be because we have villi here, then you should know that the cells on the outside are simple columnar. These two villi here are cut in lengthwise. This one shows a lacteal. The lacteals absorb lipids. Now, lipids can only be emulsified, they can't be fully digested. They can be incorporated and used for an energy source or stored when they get to a very small size. The lacteal gathers the lipids. Serving as the circulatory supply in and out within the villus is the lamina propria. Not labeled as the third part of the GI tract here. And this is the tunica, I'm sorry, not tunica, but muscularis mucosa. Muscularis mucosa. Okay. The most outstanding feature here is the large intestine, but we first need to track back to the small intestine, find where it exits. Okay, exits right here into the large intestine, and that would be the ileocecal valve, from the ileum of the small intestine into the cecum. Both are part of the GI tract. Okay, here's the cecum here. Attached to it may be the appendix, also known as the vermiform appendix. We seem to have no function that we can bet on anyway for the appendix. First part then of the large intestine is the cecum. The food, or partially digested food, I should say, moves upward along the ascending colon. It's helped along by being compartmentalized in these haustra. The haustra form all the way through, and they're really an outstanding feature of the colon. And they form because of three bands of longitudinal smooth muscle. There's one on the back side, there's one on the front side. You can't see those. You can never see all three at the same time. Some are always hiding from you, like this one going over the back. The Yersinia coli contract not at the same time, but asynchronously, and by doing so, they form these pouches. This turn right here, first turn from the ascending colon, this is the hepatic flexure, then the transverse colon, the splenic flexure, the descending colon, and then when it enters into the area of the true pelvis, we call it the sigmoid colon. Function of the colon. You gotta know, when you say any of these, try to remind yourself. I always try it. Sometimes I forget too. But what's the function? reabsorption of water, and compaction of waste. Okay, well, here's a leg, okay, and we can definitely, we got to sort of take this one apart. I know it's labeled, so you don't really have to, but if it were unlabeled, you would look and say, well, there's small intestine. That's for sure. That's the small intestine. Here it goes here, and it goes into this larger structure. Okay, this is the ileum, ileocecal valve, large intestine here. In the cat, the intestine goes essentially back up to the diaphragm, sort of makes a quick turn and comes straight back down. The liver, we have it here in lobes. Since it's lobed, it has to be a gland. It's the largest gland in the body. And since it's formed from glands, a glandular tissue, then it must be formed from glandular epithelium. Again, okay. Name in your mind, if you can, some functions of the liver. There are 605 known functions of the liver. Give me one.
Detox.
Detoxification, yes, that's a good one. Detoxification, okay. Purifying the material that's taken up by the small intestine through the hepatic portal system. The production of bile.
Bile,
We talked
yeah.
about that as well. And the other function would be the control of body temperature and homeostasis. Also, it serves as the repository for glucagon. That's extra sugar. I'm sorry, glycogen. It holds glycogen and stores it overnight until you're ready to use it again in the daytime. Okay, or any time that you have too much sugar in the blood, it will try to lower that by storing it as glycogen. Doesn't mean it's a cure for diabetes. No, that's way too much sugar. Here's the gallbladder. We know its function, I hope. Here's the diaphragm, prime mover of respiration. It's really a nice picture when you look at it. Okay, here, the small intestine is embedded in this, but this is a sheet of tissue called the greater omentum. The greater omentum. Where's the stomach? It's buried in there too. Can't see it. This is a freshly opened one. Okay, now if we've gone over this one before, great, because it always bears reviewing. If we haven't gone over it, you'll tell me at some point, and I'll go over it again. I may hold a review session in the discussion area sometime that you wouldn't be required to attend, or you could watch on the video that we have. Okay, but that's for the distant future. We just really started this section. Okay, here's a liver. You're looking at it from the top down. Nope, that doesn't make sense. Give me a moment. You're actually looking at it from the bottom up, as if you could lie on the ground and look straight up, somebody stand over you, and here's their liver cut, okay? Because this gallbladder is going to be on the right side, okay? On the right side, and it'll be towards the front. That can only happen if I take this here, this part, and pull it back down and push this to the front. Okay, don't worry about that. I'm going to ask you that. I'm just trying to get the orientation straight, and I hadn't even thought of ever doing that with this model before. What I usually do with this model is just like this. Start with what's easiest. We know it's the liver. Here's the gallbladder. Okay. They always make the gallbladder green. Okay. And we can see the cystic duct right here. It's not labeled, but that's the cystic duct. Okay. Here are ducts here and here. These are hepatic ducts, and they join the cystic duct here, right here. This forms the common bile duct, and it's not labeled. Okay, let's get it straight that we know that coming from the liver, bile is produced and delivered by the hepatic duct towards the small intestine and the ampulla levator. It will back up when there's too much to conserve it into the gallbladder and enter through the cystic duct. When it leaves, it also exits through the cystic duct. So cystic duct, hepatic duct, common bile duct here. Now, dead center. Okay. And that's looks like baby blue to me. Okay. Let me call it that. This is the hepatic portal vein. This is coming from the small intestine, not just one little part of the small intestine. The entire small intestine has blood vessels, a great circulation. And when it picks up material that it has gotten through digestion and absorption, that blood is considered contaminated still, unprocessed. And it comes to the liver through the hepatic portal vein. And then it is spread out so that the liver can share the responsibility of cleansing it anywhere you see this baby blue. Okay. And that's what happens. Then it goes to the lobules that are going to show up here in another slide in just a second. Okay. And they detoxify. Once the blood has been detoxified at least one time, it will go to the inferior vena cava. It will go out through the veins that are shown here. So you have blood in need of decontamination or detoxification, getting that process done by the liver, and then that cleansed blood leaving and entering into the circulatory system by the inferior vena cava. The liver itself has to have its own supply of already detoxified blood. Where did it get detoxified? By being at the liver earlier on a different circulation, a different time. So clean blood comes in through here. This is the hepatic artery. And it spreads out to nourish the entire liver, all the red. And when it finishes delivering its nutrients and oxygen, it exits also by the veins that we saw earlier and exits through the inferior vena cava. So there's two blood supplies to the liver, the hepatic portal system and the arterial system of our regular systemic circulation. That's through the hepatic artery and through the hepatic portal vein. Blood comes two ways, goes out only one, goes out through the inferior vena cava. Now we saw a slide of this, but I think seeing the diagram helps out considerably. This is a slice of liver, and this thing here that looks like a bit of, or is it Braunschweiger liverwurst, stuff that I don't eat, but I've seen it. That is a lobule. That's the fundamental unit, or the physiological unit of the liver, is the lobule. Okay. Maybe it looks like a grain silo. Now, there's another one here cut, another one here cut short, another here short, another here. But if they were all standing side by side uncut, they'd look identical. There's a central vein in each lobule. Here it is here. Here it is here. And the blood coming from the hepatic portal system from the small intestine has to get up here and get to this glandular epithelium to be processed. Well, we have a better picture for that right here. Here's that central vein. That's our destination. But the blood comes in from the small intestine, from the portal vein. And then the blood comes through these small channels, getting smaller and smaller, the size of capillaries. And it is processed or detoxified, if it can be. There are some toxins, of course, that are lethal immediately. And then the blood, having been detoxified, enters into the central vein. And all the central veins eventually come together into other veins that empty into the inferior vena cava. This area here has three structures. Here's the portal vein, a piece of portal vein. Here's the hepatic artery that's come this far to nourish the liver. And this is a bile duct that comes from the liver itself and is making its way to the hepatic duct to join with the cystic duct to form the common bile duct. There's 1, 2, 3 things. We call that a triad. This is a triad here. This is a triad here. Anywhere 3 of these guys come together, it's a triad. There'll be 6 triads around each lobule. Okay, here's 1, 2, third one doesn't show here, 4th, 5th would be out here, 6th would be here. Figure a cross section of the lobule like this one here is roughly a hexagon in shape with 6 points, 6 corners, and each has a triad. This little summary here shows the important types of tissues in the digestive system. Glandular epithelium for the parotid glands, the liver, and the pancreas. Stratified squamous for the upper esophagus, and then the rest of the GI tract is all simple columnar epithelium. Okay, our last thing to do today is take a look at some more tissues. Okay. These are connective tissues. it's sort of self-describing. Connective tissue connects one thing with another. It can connect two things that are the same together, or it can connect two things that are not the same together, such as muscle and epithelium. The first type of connective tissue is a relar connective tissue. I have never had a person miss a relar connective tissue when it's on a test. It's just uh, uh, so unique. Okay, you see the nuclei inside of the cells. They're surrounded by a matrix, which is invisible in this rendition. It looks like you've plowed into a wall or a bush, and that's your windshield. A relar connective tissue can be found in most places where there is a lot of flexibility. For instance, skin around your neck, it usually is quite flexible. Adipose tissue or fat. We'll have this one on the practicum for sure. It's an easy one. It sort of looks like soap bubbles. Some people see it as floating marshmallows, but it's clear. There are nuclei, but they're usually small and peripheral. Fat stores energy. Fat guards against trauma and insulates the body. Here it is around a vein. I showed you this picture, I think, before, but this is all fatty tissue here, adipose. In any slide, there's probably going to be some fat. Even though we may be asking something else, there's probably going to be some adipose. Dense regular connective tissue would be represented by tendon. Tendon, think about your Achilles tendon, for instance, it's dense. Regular means it runs in one direction. All the portions of it run in one direction, and it connects. Obviously, a tendon connects muscle to bone. Here it is, cut. Now, you may want to look at this one and say, it's wavy. It should be straight. In real life, it is straight. It is straight, and it's not bad even here. But in cutting it with the microtome, a knife used in making histological preparations, you're going to compress the tissue slightly, and the waviness is what we call an artifact. Can't be helped. Dense irregular connective tissue. Before we get to the practical exam, I will say this, I'm sure, 4 or 5 more times. The dermis is dense, irregular connective tissue. Here's the epidermis of the skin. The epidermis is stratified squamous. Okay, it's not in today's chapter because it was in Tuesday's chapter on epithelia. So here's the epidermis, here's the dermis down here. It's even got some sweat glands embedded in it. But this particular slide does not have oil glands, just like parts of your body don't have oil glands, like the palm of your hand. Okay. This is dense, but definitely irregular. I think in learning these, the trick is not to take apart each slide and say it looks dense or it doesn't look dense. It looks irregular. It's not irregular. The trick is forget that. Learn what the slide is. Learn that this is skin. Learn that this is dermis and have it memorized. That the dermis is always dense, irregular connective tissue. Okay, and then you got your answer immediately. The bone that we've looked at before, okay, compact bone here. Here's the Haversian canal in the center. Yes, it looks lopsided, but that's just the way it is. It's compressed. They're all trying to uh, push in, find their own spot. The dark spot here would be a lacuna. We would know, but we cannot see that there's an osteocyte inside each lacuna. Here's a lamella, a lamella, a labella, another word for layer. Okay, here's nicer. This is cancellous bone, also known as spongy bone. This sort of borders on being a reticular type tissue because it's got caverns and caves and nooks and crannies, if we call them that. Also in bone, you have the formation of red blood cells and your white blood cells and your platelets. All of your circulatory system is born in the bone. Well, if you would imagine a femur, okay, either from our bone kit that we used or anything else in your past, the diaphysis or shaft of the femur is compact bone. But at the proximal and distal ends of the femur, if you were to cut into it, you don't have this circular pattern. You have this pattern here of spongy bone. Please do know it by both names, cancellous and spongy. Here's hyaline cartilage. Every slide I've ever seen of hyaline cartilage looks different from the next one, and they can be a different color depending upon how they were stained. But there's just something about hyaline cartilage that always gives it away. It is the #1 type of cartilage that we have in our body. You find it in the nose, you find it in the cartilaginous C rings. Okay. And here is the nucleus, and surrounding it is the matrix. Matrix means what it's embedded in. Where did the matrix come from? The cells themselves have produced it. Okay. So when we think of cartilage, we think of things like, oh, a chicken breast, and you see the bone inside of that, or some cartilaginous fish will show it for us as well. Okay, and we can again wiggle our nose, and that's hyaline cartilage. Cartilage of all types is avascular. I'm going to show you elastic cartilage next, but right now I'm going to say that all cartilage is avascular. That means no blood vessel penetrates it. So it usually cannot be very large because it has to be able to get nutrients to all of its cells. All cartilage is avascular. Inside of the compact bone, we find in the lacuna the osteocyte. In cartilage, this cell is called a lacuna also. And inside it is a nucleus, but we call it a chondrocyte. Chondrocyte. CHON being the shortened version of cartilage. I look at this and I see people standing very close together, very crowded together. And I'm looking down from a drone on the shoreline, and people are standing in the water. But sometimes it takes that till you got it down, but you can dream up something that things look like. For instance, here's one. This is elastic cartilage. Elastic cartilage is in your ear. That makes sense. Bend your ear down, it pops right back up, fortunately. Okay, so elastic cartilage is there. Another place that we have elastic cartilage is in the epiglottis. The epiglottis is not hyaline cartilage, it's elastic cartilage. I don't think it would last long if it were hyaline cartilage. Now, if you look at it here and you think, well, it's got to be that band across the center. But is that a band? What's the real three-dimensional shape? You'll only get that if you're on your own by looking at a book, and that'll help you. But I'm here to help you right now. Think instead of a wall of stone, because it does look like that a little bit. Think instead of a pencil lying across the screen. The pencil is round. Okay, it goes all the way around. Okay, but the pencil does not go on indefinitely towards the back or towards me. It's surrounded instead here by a lot of tissue. This is more connective tissue. But the job of the elastic cartilage in the ear and in the epiglottis of changing shape and then bringing it back is what's important. Okay, so this piece of elastic cartilage that I have here, again, does not go on indefinitely. It is really more or less like a pencil shape here, or in your ear, you can imagine the shape that has, it's a little bit more convoluted. I've got my website here once again, and let's see where it takes us. Okay. Don't know why this always comes up so small on my screen, but it's easy to enlarge. These are the various types of tissues that you're responsible for. You're not responsible for anything in parentheses. Okay. So over the weekend, if you didn't last weekend, you can begin to look at these. You should be familiar with all of those in the first half. Earlier in the semester, we looked at skeletal, cardiac, and smooth muscle, and we've looked at the neuron. So you really shouldn't have difficulty with that. But if you do, give me a shout. Then on the organs, we've looked at the salivary gland. We've looked at the tongue, in the esophagus, in the stomach, small intestine, large intestine. We've actually looked at all of these once. What you're going to need to do is look at it, different slide, okay, and say, that's the lung. What's the function? And you scratch your head. Well, everybody knows the function of the lung. Why should I have to know? What's he really asking? He's really asking, what is the function of the lung? Okay, so respiration is all you would really need to say there. But what's the tissue type that's prevalent here? And that'd be an arrow pointing to it. And it's very thin like this right here. That's simple squamous. Okay. Now we will be going over all of these again, probably again in the review session, because some people will need it, or some people will think they need it, and some people that want it to be sure that they got it down. Okay. Now, any questions?
For Charlie, for final exam, it will be 50 questions for lectures and 50 for lab.
No.
Then
You have on the final exam day. I'm. Is that the somebody with the syllabus right in front of them? Is that the 16th of December?
I think it is, yeah.
Okay. The day before that, you have L3. That's the material that we're talking about right now in lecture. That's the autonomic nervous system, the nervous tissue. It's also the integumentary system, because we didn't include it on the previous exam. I'll make all this clear to you in plenty of time. First, we have L3. Then on the next meeting, I put it off until finals week. Normally, we would have the practicum earlier, but I want to give you the extra day to study. And since we're online, not anybody's way, you know, I'll have that one on the 16th, like I did with my last two classes. The first thing you'll do is you'll take the practicum number 4. Okay. Get that out of the way, and then you'll go into at a set time. It'll open up, and you take the final. The practicum is 100 questions, as it always is, and presented just like it always is. I have lots of pictures this time again. The final examination is 50 questions. Okay. Mainly because, well, we don't need 100 questions on the final to see that you still do or do not remember the material from the beginning. And then I'll show you the list of terms again, which I've shown you before, and you can easily get from my webpage, list of terms under lecture topics at the very last. And so far, we've used 2/3 of those terms. The other third are going into the third lecture exam. But then for the final, okay, and for the final, all of those terms come into play. Okay. But I asked 50 questions. I asked a multiple choice question, and I have 4 or 5 answers for you to pick from. And those 4 or 5 answers are all terms from the list of terms. The list of terms are the answers, okay? If you know which one to pick, of course. And that's it. Most people finish that final in 20 minutes. Okay. You'll have longer than that. Okay. If you're not convinced yet that you know or believe what I said, it will come to light that, yep, he said exactly what he's going to do, and it's exactly the way it came about. And I'm not expecting to get any people with special difficulty with that, because I never do, never do. Okay. Okay, watch for a quiz sometime this weekend, and it'll be on the nervous system, and so it may take a little bit more thought, but watch for it. Okay. And then otherwise, have a good weekend and see you on Tuesday.
Thanks,
Bye-bye.
Charlie.
Bye.
Yeah. Thank you.
Thank you.
Thank you.
Bye-bye.
Thank you, Charlie.
Bye-bye.
Bye.